Hi everyone. This week we're going to be talking about genes and genetic diseases. I will first do a bit of review of any normal anatomy and physiology that's relevant, and then we're going to be talking about different types of mutations that exist in the body as well as abnormalities of chromosomes. Now, when it comes to talking about genes and genetic diseases, what is genetics? Well, genetics is a study of biologic inheritance. And genes, which are composed of DNA, are those basic units of inheritance. And having an understanding of genetics is really essential to understanding human life and can therefore help understand human disease. To date, there are, probably well over 20,000 different genetic traits and diseases that have been identified and cataloged. Great progress is certainly being made in the diagnosis of genetic diseases and in the mechanisms that underlie them. There's, for example, gene therapy. Gene therapy is the utilization of normal genes to correct genetic disease. So that's a very intriguing advancement in medicine. All right, first, let's do some review. I won't be specifically testing you on normal anatomy and physiology, but having the background is important because components of normal anatomy and physiology will be incorporated into discussions of abnormal or altered physiology. So before we dive into genetics, let's do a quick reminder of what DNA actually is and why it matters. So DNA and RNA are both nucleic acids, and they're built from these repeating units that are called, so the DNA is built from these repeating units that are called nucleotides. And a nucleotide has three parts that make it up, and that's a sugar, a phosphate, and a nitrogenous base. The sugar and the phosphate here form really what's called the backbone of the molecule, and then the nitrogenous bases are what actually carry the information. Now, when we talk about DNA, there are four nitrogenous bases: adenine, thymine, cytosine, and guanine. And in DNA, these nitrogenous bases are going to pair in a specific way. Adenine will always pair with thymine, and cytosine will always pair with guanine in DNA. And it's this specific base pairing that allows DNA to form that characteristic double helix that you can think of looking like a twisted ladder. So let's see here. You can see the backbone here, which is the sugar and the phosphate. And you can see the pairing of adenine and thymine and guanine and cytosine. And it gives it a ladder look. So if we were to sort of draw that out untwisted ladder, what we have is the rails of the ladder here are made up of the sugar and the phosphates, whereas the rungs here are made up of, let's say, this nitrogenous base pairing with this nitrogenous base. These are the rungs of the ladder, this nitrogenous base pairing with this nitrogenous base, and so on and so forth. Now, RNA is closely related to DNA, but there are key differences. RNA, first of all, is usually single-stranded versus double like we see with DNA. And RNA uses uracil instead of thymine. So in other words, thymine is found in DNA, but not RNA. And uracil is found in RNA and not DNA. And this difference is important for how information is copied and used in the cell. Finally, DNA has this unique ability to replicate. You should know that by now, that DNA can replicate. During DNA replication, what we see is that the two strands separate. So here is intact. Here the DNA is all coiled up. And then here it's separated. So normally, if you take away the orange part, it was the blue or purple color that was together tightly wound to form this. So during DNA replication, the DNA or the strands of the DNA are going to separate. So you can see that this one has separated from this purple one. And so once we've exposed, as you can see, these nitrogenous bases are now exposed because the DNA has unwound. Because of the specific DNA sequencing that we know exists, we have all of these free nucleotides that will find their match, bind with it to form two new strands of DNA. So you can see here what we've formed. We have a nucleotide, they kind of float in and they're going to bind with their appropriate match. You can see that's happening here, adenine bound with thymine, cytosine with guanine, and that will keep happening. And what we end up with is replicated DNA. And DNA polymerase is an enzyme that plays a big role in that. So again, because of that specific base pairing, the sequence can be copied accurately, which is essential for appropriate cell division. It's also appropriate for growth and repair. So at its core, DNA is this really stable, self-copying molecule that we know stores genetic information. And then RNA, that's involved in using the information found within the DNA to make proteins, remembering the concepts of translation or transcription and translation, which I will elaborate on soon. So that ladder that I drew earlier, you can see this looks like a ladder here. This is representing a very simple image of non-spiraled DNA and making it look like a ladder where we have the sugar and the phosphate that make up the rails on the side of the ladder, and then the nitrogenous bases with their appropriate pair making up the rungs of the ladder where you would step and put your hands on if you were climbing the ladder. And so we would call this here a nucleotide, and this here a nucleotide. So I'm including there the sugar, the phosphate, and the nitrogenous base. And so the nucleotides then are really the basic building block of nucleic acids, which are DNA and RNA. A little bit of a reminder on the cell cycle. So the cell cycle is, as you would guess, the life cycle of a cell. How it grows, copies its DNA, and then ultimately divides if it's a dividing cell. A few things to point out and remind you of is that most, and this shown in this green color here, most of a cell's life is spent in interphase. And interphase includes G1, that's really a growth phase, S phase, where DNA replication occurs, that's the key feature there, and then G2 phase, which is kind of the final preparation phase, getting the cell ready to enter cell division, which is the reproduction phase. And when I say reproduction, I mean taking one cell and making two identical cells. So when the cell enters this cell division phase, it's going to go through mitosis, involving prophase, metaphase, anaphase, and telophase. And then the final component is cytokinesis, which is the final division of the cytoplasm, which will form two separate cells. Now that's true of cells that are dividing cells. Some cells, what they do is they enter G0 phase. This is where they aren't dividing. They just focus on normal function. And this is very common in neurons and muscle cells. So this is just maintenance. Apoptosis, a term that will come up quite a bit. This is defined as programmed cell death. It is this controlled and intentional process where damaged or unnecessary cells are safely removed. So it's not accidental. This is a protective mechanism. If a cell is abnormal and we can't fix it, we need to send that cell to death. Otherwise, problems will ensue. An abnormal cell, let's say this is an abnormal cell. If we don't fix it or send it to death, then when it divides, this is just an example, it produces now two abnormal cells. And then this keeps going and going and going. It can lead to, for example, the development of tumors, the development of cancer. Okay, so anything that goes wrong in the cell, let's say there's a mutation of DNA, and the cell cycle is interrupted or apoptosis doesn't exist, it can lead to disease. So genetic diseases often come down to problems with DNA replication. So something goes wrong with replicating the DNA. Something maybe goes wrong with cell division, or maybe there's a problem with programmed cell death. In any case, it can lead to genetic diseases. All right, a very important role that cells have, as you might recall from normal anatomy and physiology, is the production of proteins. And remember that the instructions on how to make those proteins come from the DNA that's found within the nucleus. And where do we make the proteins? Well, remember, proteins are made at ribosomes, whether they're attached to the endoplasmic reticulum or free floating in the cytoplasm. In any case, the instructions on how to make that protein are found within the DNA, but we can't take the DNA out of the nucleus and bring it to the ribosome. It has to stay within the nucleus. So instead, what we can do is take a copy of it and take that copy to the ribosome. And that leads us then into talking about transcription and translation. So on this slide, we can see the two major steps that are involved in gene expression. So really looking at how information found in DNA can be turned into a functional protein, because so many things in the body are proteins. So we need to be able to make these properly. The first is transcription. Transcription happens in the nucleus. So this is representing the nucleus here. So transcription occurs in the nucleus. Now, during transcription, a specific segment of DNA is going to be used as a template to make messenger RNA or mRNA. So what do I mean by that? Well, let's say the instructions to make a particular protein are found, for simplicity purposes, this is all DNA here, right? Let's say the instructions to make a particular protein are found, let's say right here. We need to unwind this instruction, this area of the DNA, to know how to make the protein. And then we take a copy of it from there. So it would be like, let's say you wanted to make banana bread and you have this big recipe book, going old school, and the recipe, you don't need the whole book to make banana bread. You just need a particular recipe. And if you wanted to, instead of carrying that big book with you somewhere to make the banana bread, you can transcribe or write out that recipe on a separate piece of paper and then take that paper with you to wherever it is that you're going to make that banana bread. So what we need to do then in the nucleus is take a copy of this relevant segment of DNA that has the instructions on how to make whatever particular protein we're talking about, and then make a copy of it, and then take that copy out of the nucleus to the ribosome where it will be made. So again, during transcription, a specific segment of DNA is going to be used as a template to make messenger RNA. So that's the copy. Okay, it's called transcription because you're transcribing a copy of it. And the process is carried out by a slightly different enzyme. We talked about DNA polymerase on an earlier slide, but RNA polymerase. You can see that here's the normal DNA. It's been unzipped in a particular area that contains the instructions to make the protein of interest. And you can see now an mRNA strand is forming by taking a copy of what exists here. And so what we end up with transcription is this mRNA, this copy, messenger RNA, it's carrying the message, and it's complementary to that DNA sequence, with the only difference being that we have uracil instead of thymine, as talked about earlier. Now, after transcription's done, after we form mRNA, there's a lot of things that are happening where it's sort of cleaned up and made how it needs to be before it leaves the nucleus. Once it's ready to go, so here's mRNA here, it's going to leave the nucleus through nuclear pores, and it's going to find the place where proteins are made, and that would be the ribosome. And that's where we get to the point of translation. So first we transcribed a copy of the DNA, and now we're taking that piece of paper with the banana bread recipe to the place that we're going to make it. And we're going to translate that recipe into an actual protein or banana bread. Now the ribosome is going to read the message, read the messenger RNA, and it reads it 3 bases at a time. So let's highlight that. So here is 1, 2, 3, 1, 2, 3, 1, 2, 3. These 3 nucleotides that are together are sometimes called the 3-base sequence, are sometimes called a triplet because there's 3 of them, or more appropriately, a codon. And each codon, so group of these 3 base sequences, specifies for a specific or a particular amino acid, because we know amino acids are the building block of proteins. So what happens is we have this transporter called tRNA, or transfer RNA. Its goal is to find amino acids, and you can see one is bound to it here. And I'll just erase the slide so you can see, this amino acid is bound here. It finds an amino acid, and it's a specific amino acid. It's a specific amino acid that really matches with this part of the tRNA here. It's called the anticodon. So think of it like a plug. We have tRNA matches with the codon from mRNA, and they fit together like a plug. So this here is going to fit, you can see it getting bigger, very specifically with this here. And it's only going to match appropriately. So if this is a codon that codes for this specific amino acid, then it's only this transfer RNA with that amino acid that will bind with it. And then little by little, you'll be joining together all of these different amino acids, linking together via peptide bonds. And the chain will get longer and longer and longer. And then it'll eventually fold and take a certain shape to become a protein. So really, a summary is that we know DNA stores the instructions, transcription copies those instructions into mRNA or messenger RNA, and then translation uses that mRNA to build a protein. And this whole flow of DNA to RNA to protein, this is also a central concept to understand when we talk about genetic mutations and how it affects cell function and leads to disease. We need to produce correct proteins. If we produce wrong proteins, problems will occur. And actually, I didn't reference this diagram here at the top, but this is a good example to show you that this is transfer RNA. It has a couple spots that will bind to the ribosome to keep it there. It has its binding site for a specific amino acid, the building block of a protein. And then here is the sequence that will bind to the bases of the codon at the ribosome. Now that's normal anatomy and physiology. So now we're going to see what happens when things don't go normal. They're not the way they should be. And we're starting talking about mutations. A mutation is really any change in genetic material. And these changes can be things that you inherit, so you get from your family, or they can be acquired over time based on maybe things that you're exposed to. So that means you can either be born with a problem, a mutation, or obtain one at some point throughout your life. And it's important to note that many mutations are detected and corrected by our built-in DNA repair system. In other words, not every mutation is going to cause a problem. We do have these repair systems in place that can fix the issue so that it's not a problem. I want you to think of it like this. You have this big recipe book and you want to make banana bread. And you open up the recipe book to the page on banana bread. But somebody must have spilled water on it or part of the page is ripped and you cannot read the instructions properly. But you're like, I'm still going to go ahead and make the banana bread. But you didn't do it right because the instructions were not provided to you properly. And so the banana bread did not turn out. And so that's what happens with our DNA. If there's a mutation, if there's something wrong and we can't correct it, then what happens is we produce abnormal products. It's like reading the recipe is wrong. There are several types of mutations that exist. So we have base pair substitution that exists as a missense and nonsense mutation, frame shift mutations that involve base pair insertions and deletions, spontaneous mutations, and chromosomal aberrations. I will be elaborating on these primarily, I'm spending quite a bit of time on these, but we'll talk about all of these on upcoming slides. Now, if we know a mutation has occurred and the body can't correct it, what are some of the effects that you might see? Well, not all mutations are going to have the same effect. There's going to be quite a spectrum of potential effects, but we can group them into three main categories. Some mutations are silent, meaning that they don't affect cell function and they're clinically undetectable. These are actually quite common, so they're not a problem most of the time. Other mutations are considered sublethal, meaning that they do disrupt function, they do cause disease, so they're not silent. And then some mutations are lethal, also not silent, but lethal ones prevent normal development or survival. Finally, mutations can be caused by what are called mutagens. Mutagens are just agents that are known to increase the frequency of mutation, and this can include things like exposure to radiation, exposure to chemicals, even exposure to certain biological agents such as viruses. So mutations are common. They vary significantly. Most of them are harmless, but some can significantly impact cell function and lead to disease, and so those are the more relevant ones that we will be drawing on mainly for this course. And this section of the course especially. So what we're going to do first is look at base pair substitutions, missense and nonsense mutations, and also frame shift mutations on this slide. And what I've done is this is a removable text box, but you can see that first we're going to look at missense mutations, then we're going to look at nonsense mutations, and then we'll look at frame shift mutations. The first thing I want to point out to you on this slide is just to note the normal DNA. So this is a simple version of DNA, but we've twisted it compared to how we looked at it unspiraled on an earlier slide. Now it's a bit hard to see and might make you feel a little bit cross-eyed trying to follow these different bases. But if this is normal DNA and this is normal mRNA, you can see the copy that's made. So for example, if we look at these three guys here, this would be a codon, C, G, G. A codon, again, is just a group of three. And if we were to take a copy of CGG, the copy complement would be GCC. GCC, in making a polypeptide, codes for this amino acid. And this amino acid, this is just the short form for alanine. So I'll say it all again. This is normal DNA. If we were to take a copy of it, so a particular section of this DNA, we want to take a copy of this in order to make a certain protein, this would be the copy. And if you follow it along, just starting at the beginning for simplicity, CGG, the copy there is GCC. GCC, that's a codon group of three, codes for a specific amino acid, which is alanine. So if you think back to, let's go to over here for a moment. Let's say this codon here on mRNA that I just described codes for alanine. That means that transfer RNA is going to specifically bring alanine to this section of mRNA at the ribosome. So it can, as I mentioned, make you a little bit cross-eyed trying to line all of these up. But what I want to point out is that these three here, as part of mRNA, code for this amino acid. These three here code for this amino acid. These three for this one, and so on and so forth. So what we're doing is we're linking all these amino acids together to form a polypeptide, which can form proteins. We need this specific order of amino acids to form the particular protein of interest. If anything here changes, that's a problem. Of course, this course is all about problems. So let's look at an example where normal does not occur. And that is with what's called a base pair substitution. So substitution of a base pair, which is called a missense mutation. And what happens here is we get a change in one amino acid in the production of a polypeptide. So one of these guys changes. Let's see what that looks like. So let's start up here. This is what is normal, GC. What we're going to do in this base pair substitution is substitute AT in for GC instead. So now you can see we have AT. So adenine and thymine now, we have created a base pair substitution. So what happens? What does that mean? Well, let's use a pen. I'm going to circle this. So GC normally, when mRNA takes a copy, will have G, right? And then we know that these three, A, G, C, code for this amino acid. If we get a mutation here where adenine and thymine are now inserted here instead of guanine and cytosine, now what we have is a different copy is made. So now we have A because it complements T, whereas before we had G, which complements C. AAC codes for a different amino acid. So now you can see that in the formation of this polypeptide, there's a different amino acid, which means we're going to form a different polypeptide. So when we talk about a missense mutation, we have one amino acid in that production of the polypeptide that changes. In this case, it's shown here, all because of a different pair, different base pair being put into the DNA and kicking out GC. It is possible that this will not create any effects clinically. So it is a mutation. But because of some redundancy of the genetic code, there may not actually be any consequences. Or there could be profound ones. Next up is a nonsense mutation. When we talk about nonsense mutations, it's still a base pair substitution like we talked about here. But this one inserts a stop codon, so a group of 3 nitrogenous bases, inserts a stop codon to produce a shortened polypeptide, and that shortened polypeptide from normal may lose its structure or function. So what does that mean? So we've got the same DNA strand that we're looking at, the same normal one, we're just looking at a different section. And here what we're looking at is CG, this base pair, in normal DNA, and then the copy and match with guanine is cytosine. And then these 3 here, this codon UAC codes for this amino acid, tyrosine. So with a nonsense mutation, we're going to do the same sort of substitution that we talked about here, where adenine and thymine are going to be substituted into where cytosine and guanine were. So what that means is with this mutation, we're kicking out CG and instead inputting AT. So you can see everything else is the same, except we've kicked out CG as a base pair and inserted AT. Then when the mRNA takes a copy, well, now it's going to match the thymine with an adenine. Now our codon, which was this that codes for this, is now UAA. UAA doesn't code for any amino acid. UAA, UAG, and UGA these three groups of codons are called stop codons. If these form together, these form a codon, like we can see here with UAA, it stops the formation of the polypeptide right there. So instead of having, in this simple example, 5 amino acids to form a polypeptide, now we only have 3, and it stops. You can see nothing happens after that. So we end up with a short polypeptide compared to what it should be. So these stop codons really then terminate the translation of the polypeptide. Next up is another form of mutation that's called a frame shift mutation. And frame shift mutations, as you might remember from this slide here, include base pair insertions and base pair deletions. So it's not a substitution. We're not removing a base pair and substituting it for another one, like we saw with these two examples. Removing a pair, substituting with a pair. Removing, substituting. Instead, we're deleting a base pair completely and not replacing it with anything, or inserting an additional base pair without removing anything. And so as you can probably imagine, if you're inserting or deleting, it's going to cause an entire shift in the reading frame of that messenger RNA. So what we end up with is a different polypeptide, which affects structure and function. So let's see what that looks like. So what we have here, again, is that normal DNA, the normal mRNA, and then the normal polypeptide composed of the amino acids based on the specific codon. What we're going to do with a frame shift mutation is we're going to insert 2 base pairs. So we're not removing anything, we're just inserting. So let's look here to see what we would normally observe is, I think it's this area here. You can see that we have thymine, adenine, adenine, thymine. We're going to now insert both an adenine and thymine pair and a cytosine and guanine, which you can see here. So we've inserted them in between these. So this, as you can expect, is going to shift everything because we've shoved, base pairs in there that normally shouldn't be there. And when that happens, it changes everything. Now we're going to take a different copy. CAA now codes for this amino acid instead of what we would see up here. And it shifts everything forward. So now we have a different group of 3 copied and another different group of 3, because if you follow it along, everything changes downstream. And so we end up with all different amino acids forming the polypeptide. So it's really a whole different polypeptide than it was supposed to be. So these mutations change the entire reading frame of the DNA sequence because the deletion or insertion is it's not a multiple of 3, first of all, and it creates an entire shift in what amino acid sequences are present and therefore producing a different polypeptide. So this is still frame shift mutation, just to show you a different way to look at this. This is representing DNA. This is separated, showing you the different groups of 3 that exist. And if we remove this and delete it, okay, so removing this completely and deleting it, everything shifts. The group of 3 shifts. So what we saw once as this group and this group and so on and so forth is now completely different. And the same thing would be true then with an insertion. If we add in another base pair, you can still see that from the original, everything now is shifted and we have a new group of 3. You can maybe think of it as, let's say there's a whole bunch of people and you're being broken up in groups of 3 and standing in a line. And the people that you're standing with in that group are who you're working on a project with. And then all of a sudden, somebody leaves that group. Well, or you have more people randomly joining in the different groups. Who you're partnered with and grouped with is going to change. All right, now let's look at some of the effects that you can see with a base pair substitution. Here's a list of the amino acids in the body. Make sure you memorize all of this. I'm just kidding. Don't memorize any of that. Let's look at a situation that's normal first, because then it helps us look at different types of mutations that can exist. So we're going to compare normal then to a silent mutation, meaning it doesn't have any clinical relevance. And then we'll compare normal to a not silent mutation, and therefore clinically relevant. So we're looking at things similar, but just a bit different than how we did on the last 2 slides. Here is normal DNA. Here is normal, let's call it mRNA. This is your messenger RNA. We know that if we were to unzip this and take a copy, that mRNA would form the complementary match with these 3, right? C with G, A with U, because it's RNA, A with U, and then this group here, when we unzip it, take a copy, G with C, T with A with U, and so on and so forth. Remembering that these 3 here, groups of 3, are called codons, and a codon on mRNA is going to code, again, as a reminder, for a specific amino acid. You can see that all the way through. These groups of 3 code for specific amino acids. And so when we produce a normal polypeptide, it means that the amino acids are all where they should be in the correct order. Now let's see what happens with a silent mutation. In this example, we're going to look at this, what was GC here, and therefore the complementary copy for mRNA to C would be G, and then we end up forming these 3 together, UUG, which codes for leucine. With a silent mutation, and by the way, remember we're looking at base pair substitutions, right? Which bring us back to missense and nonsense mutations, which were the first 2 here. So in this example, with a base pair substitution, if in DNA we get rid of this GC base pair and substitute it with AT, so kick this out, insert this, then when mRNA takes a copy, now it's matching with thymine, so the copy for mRNA for this section is adenine instead of guanine. So instead of UUG, which codes for leucine, we now have UUA, so a different codon. But guess what? UUA also codes for leucine. So you can see that even though there's a mutation that exists, it's okay, there is some redundancy. And we call this silent because it's minor enough that it's not going to cause a problem, because the amino acid didn't change and therefore neither did the polypeptide. Let's look normal again, but we're going to look at this example down here with AT, and we're going to see what happens when there is a base pair substitution here. So AT, if we were to take a copy, the copy of the T on mRNA would be the A, and then we have GAA, which codes for glutamate. If we kick out AT and substitute with TA, they're now in different order. Even though they're the same nitrogenous bases, they're flipped around. Now mRNA is going to take a copy based on A and match it with uracil, because we know uracil belongs to RNA. And now, instead of GAA as the codon, we have GUA. GUA codes for valine, which is a completely different amino acid. This particular mutation that is seen here is seen with sickle cell anemia. And so there's an abnormality in these cases in the red blood cell production in those that are affected. So the question I have for you is, what type of mutation is this? Well, this wouldn't be, let's go back for a moment. Which of these two would it be? We only changed one amino acid in the production of the polypeptide, so it's a missense mutation. That's the form of base pair substitution that we encountered. It's not a stop codon. Really, we're just looking at these two here because they're base pair substitutions. It's not a nonsense mutation because there wasn't a stop codon that stopped the production of the polypeptide. So missense mutation. All right, let's elaborate on mutagens. Mutagens, I defined earlier, are agents that increase the frequency of mutations, and they do so by damaging DNA or interfering with the normal DNA replication. Okay, so we end up getting changes in DNA. One group of mutagens are called intercalating agents. Intercalating agents are described as these sort of flat molecules that just slip in and slide between DNA base pairs. So they go where they don't belong. And here's an example here. This is an intercalating molecule. It intercalates or inserts itself into the DNA in between base pairs. And when they insert themselves in the DNA, this is going to distort it, right? It distorts the normal double helix. And that can make it very difficult for DNA polymerase, that enzyme that helps with DNA replication, to copy the DNA correctly. And so it can make mistakes. And so you might be able to appreciate that this insertion is sort of like a frame shift mutation. Something's being inserted, which changes the reading pattern. This is very disruptive to protein formation. And here are some chemical examples of intercalating agents. You don't need to know what these are, but this is a disinfectant, a bacteriostatic that is used to stop bacteria from reproducing. And acridine orange is just a type of dye. Okay, UV light. UV light is another important mutagen. UV radiation causes, and I'll explain this, but it causes adjacent pyrimidine bases, in particular thymine, to bind together on the same DNA strand, causing what's referred to as a pyrimidine dimer or a photodimer. So what the heck does all that mean? So I'm going to break it down and give you a few important points first. One is, what is a pyrimidine dimer or a photodimer? Well, first we need to know what a pyrimidine is. Pyrimidines, examples with DNA, are thymine and cytosine. So UV light has the ability to really impact thymine and cytosine, especially thymine. So when someone's exposed to UV radiation, the UV radiation can cause, so this is the DNA backbone, the sugar and the phosphate, can cause any nitrogenous bases or nucleotides that are composed of thymine to, and particularly the thymine part, to sort of disengage from its associated partner. So normally, let's look here, you have two thymines side by side, and they're normally attached to the adenine on the other side of the DNA, other side of the ladder. UV radiation can cause these guys to pull apart, these thymines to pull apart from their adenine pair, and instead bind to each other. So if I get a N here, we end up with these guys binding to each other. And this is shown here. We have these two thymines, which would normally be paired with adenine, right, on the other side of the ladder. Instead, they disengage from the adenine and they bind to each other. That causes a pyrimidine dimer or a photodimer. So this can cause, it's sort of like a kink in the DNA. And the enzymes involved in replication may stall or mistake it when copying it because it starts reading the DNA and then it gets to the kink and it's like what the heck is this? And this we're talking about when it comes to DNA replication. So we're trying to replicate the DNA and then it gets to the kink and it sees these thymine bases attached to each other on the same strand and it's thinking, what the heck is this? Now, cells do have a repair mechanism in place that's called excision repair. And what this does is it removes damaged DNA segments, so let's say this, and replaces them. However, the system is not perfect. It can make mistakes. Now, if damage is severe or repair mechanisms fail or let's say these mutations remain or they build up, like people that constantly spend their time in the sun, over time these mutations accumulate and we can't fully repair things properly and it can contribute to diseases such as cancer or skin cancer. I found this great image on Wikipedia showing you normal DNA and then incoming radiation, and you can see the pulling away of these bases from their complementary match. And then thymine or cytosine, which are pyrimidine derivatives, will bind to each other on the same strand. Thymine to thymine, cytosine to cytosine. Thymine to thymine are the most common. Cytosine to cytosine can also occur, and cytosine to thymine can also occur. But again, most common. Now on to spontaneous mutations. So just so that we're all on the same page, we've talked about base pair substitutions, my sense, nonsense, frame shift mutations, insertions and deletions. And now we're moving on to spontaneous mutations and we'll finish up with chromosomal aberrations. When we say spontaneous mutation, we're referring to a genetic change that occurs just by chance, right? It's spontaneous. And it's usually because of errors that occur during DNA replication, which we know occurs during S phase of the cell cycle. These are considered overall generally rare, but when they happen in key genes, then it can lead to significant disease. An important type of spontaneous mutation that we'll talk about is called trinucleotide repeat expansion. A trinucleotide repeat means that a sequence of 3 DNA bases, so let's say CGG, as shown here, cytosine, guanine, guanine. So a sequence of 3 DNA bases repeats over and over and over again in the genome. So for example, it could be CGG, 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 CGG. Now, these repeat regions are actually pretty common in the human genome and can be considered stable, but sometimes they become unstable and can continue to expand. Okay, so that's the key mechanism, the repeat expansion, where short DNA sequences repeat multiple times. The main mechanism that we really care about with this is something called slipped strand mispairing. When DNA polymerase, remember that enzyme from before, is copying a stretch of repetitive DNA, it can sometimes quite literally lose its place, especially because repetitive sequences are easy to misalign. When I say trinucleotide repeat expansion, it's really this general type of mutation that can happen in many different genes across different chromosomes. It could be autosomes or sex chromosomes. But we're going to look at an example that's specific to the X chromosome, where we see the CGG repeat, and this increase in the frequency of these trinucleotides can make the X chromosome more fragile. It becomes weaker with more repeats. So the chromosomal region or area that has the very large CGG repeat expansions becomes structurally unstable, and so it's prone to breaking. And if observing it in laboratory conditions, it can physically appear fragile at that location. Now let's have a look at this image here in this context and try to understand the progression, how we can go from few repeats to much greater repeats and what those clinical implications are. So at the top here in this simplified example, we have the normal state, or what is considered normal, where the CGG repeat region has a small number of repeats. So in this example, it's anywhere from 6 to 54 copies. So at this level, the gene will be able to function normally. No disease occurs. So this is kind of our baseline. It's considered normal. So remembering our normal, we're now going to look at NTM, which is non-transmitting male, which is a carrier of the expansion. So you can see there are more copies here compared to normal. So we can say then that a non-transmitting male is a male that carries the abnormal number of the CGG repeats. So it's abnormal now. Of course, it's found on a gene on the X chromosome. But he's usually clinically normal, meaning not showing the classic Fragile X syndrome features. Now here's a key genetics point. A father gives his X chromosome only to his daughters, and he gives his Y chromosome only to his sons. So what that means is that this is all under normal circumstances, just normal reproduction. If a male and a female have a baby, and the male gives an X chromosome, and the female gives obviously an X chromosome, then we have XX, and they have a female baby. If the male gives the Y chromosome, the female the X, we end up with XY, which is a male baby. Okay, so remembering that a father gives his X only to his daughters and gives his Y to his sons is important now, because if this non-transmitting male passes on his X chromosome, in this case, the one that is a premutation but still demonstrating those expansions, we know that he will be passing that on only to his daughter, because the daughters receive the X chromosome from their dads. Now, in many cases, if this male has a daughter and she inherits the expansion, in many cases, she may not have obvious symptoms. And that's partly because she has two X chromosomes, right? So she has two X chromosomes, this daughter, and she inherited one from dad. So one is healthy from mom, one is a permutation from dad. Now, what's interesting here, and sort of a big message from this diagram here, is that the permutation tends to stay relatively stable when it's transmitted from a male to his daughter. So meaning when a father passes on those repeat numbers, it usually doesn't expand dramatically. So this, let's call this guy Bob. Bob has a daughter and her name is Stephanie. Bob passed on those repeat expansions from his X chromosome to his daughter Stephanie, but you can see that the expansion is the same. And this is for simplicity, but really, oftentimes, there isn't much additional expansion that occurs. But here's the crucial point. When Stephanie, the daughter, as a premutation carrier, has children, that repeat expansion is more likely to become unstable during egg formation and can expand to the full mutation. So the daughter really becomes this key link. She carries the permutation that she got from her dad, and when she passes that permutation onto her child, that the repeat can expand into a full mutation, where there can be hundreds of repeats that end up leading to an affected person, so somebody with the disease. In this case, Fragile X syndrome. And so this full mutation ends up leading to abnormalities overall, leading to disease, the disease being Fragile X syndrome, which it involves intellectual disability and behavioral autism-like features. The next component of this lecture relates to chromosomal abnormalities. So we're going to be looking at chromosomal aberrations, which is really a fancy way of saying any change in the normal number or structure of chromosomes, which can often result in physical or mental abnormalities. But before we do that, we need to have a bit of reminder of what normal looks like. In somatic cells, these are the body cells, they're diploid, meaning there are 23 pairs of chromosomes, which would total 46. These pairs are considered euploid, which means they have the correct number of chromosomes present. In contrast, gametes, so your sperm and oocytes, they are considered haploid cells because they have half the number of total chromosomes, so just one chromosome from each pair, so that totals 23. And that is because when a sperm meets with an egg, their chromosomes come together, and then we end up with 23 pairs versus just the 23 chromosomes in a gamete. Most of our chromosomes, so we're talking 23 of the 23 pairs, are called autosomes. These are homologous pairs. Homologous referring to the same. This means that one chromosome comes from each parent and they carry the same genes in the same order. So this here is a computerized image called a karyotype, where we see the chromosomes all lined up here, and there are 23 total. That 23rd are the sex chromosomes, and this would be one from mom, one from dad for chromosome 1 from mom, one from dad, chromosome 2. The first 22 are autosomes. They are homologous, meaning that this from mom is identical from the perspective of the information that's carried to this from dad, and so on and so forth. But that final pair, that 23rd pair, these are the sex chromosomes. Females will typically have XX, and so we can call those homologous. They're not autosomes, but we can call them homologous because there's two X's, they contain the same information. Whereas males will be XY, and these are non-homologous, meaning not the same, because the X and the Y carry different information. And in this karyotype, we can see X and Y, and you can see how little the Y is. So this is male, very little Y. It carries relatively little material compared to the X. One last piece of normal to mention has to do with these terms at the bottom, mitosis and meiosis. How chromosomes divide matters. Mitosis occurs in somatic cells and is designed to maintain that diploid chromosome number. So each daughter cell should be genetically identical. Meiosis, on the other hand, produces gametes and intentionally reduces the chromosome number from diploid to haploid. Because meiosis involves pairing and separation of homologous chromosomes, it's also where errors are going to occur. And we'll talk about errors like non-disjunction soon. So when we talk about chromosomal aberrations, we're oftentimes talking about where and during which type of cell division something went wrong. So why all this matters is because chromosomal aberrations, coming up next, occur when there are errors in chromosome number or structure, and understanding what's normal, diploid versus haploid, homologous pairs, autosome versus sex chromosomes, allows us to recognize when something has gone wrong. Many genetic diseases arise from disruptions at exactly this level. All right. So we're going to break it up into talking about numerical first and then structural chromosomal aberrations, a change from normal. So now that we've reviewed what normal chromosome number looks like, we can talk about numerical chromosomal aberrations. And this is a situation where the number of chromosomes is obviously abnormal. These are clinically very important because they are the leading cause of intellectual disability and spontaneous miscarriage. First, polyploidy. Poly means many. Polyploidy refers to having an entire set of extra chromosomes, an entire extra set of chromosomes. So instead of 46, so the 23 plus 23, so 46 is shown here, you might see 69. So 3 of each. We call that triploidy. Or 92, tetraploidy. It's just adding additional sets, and it can be multiple extra sets. So you could have 3 of all of these. You could have 4 of all of these, 5 of all of these, and so on and so forth, as you can see with the increasing numbers, with these two being more common, the triploidy and tetraploidy. Now, while this can occur transiently, polyploidy in humans is not compatible with long-term survival, meaning that most cases are going to result in early miscarriage, stillbirth, or even death shortly after birth. So this tells us that having a whole extra genome, having all of this extra copies of the genome, is profoundly disruptive. Now, more commonly, aside from polyploidy, which is a full extra set or multiples of the extra set, more common than that is aneuploidy. So euploid. Remember euploid on an earlier slide? This is the normal, what we would see normally. Aneuploidy, and think of this as being lack of the normal amount, the normal number of chromosomes. This is more common. So we can say that aneuploidy means an abnormal number of a single chromosome. Polyploidy was an abnormal number of all of the chromosomes. Aneuploidy is lack of the normal number of a single chromosome. So in other words, we could have 3 of chromosome 12. We could have, I'm going to cross this out, pretend it's not there, 1 of chromosome 8. And the thought is that if it comes down to, is it better to have more of a single chromosome or less, it appears that loss of a chromosome has more serious consequences than too much of a particular chromosome. So better to have more than less. So this would be, in theory, a better situation. Actually, I'll rephrase that to say that loss of a chromosome is generally more severe than having extra material. And that's why when we talk about a particular type, which is autosomal monosomy, mono means 1, that means that there is one of the autosomal chromosomes. So meaning that the first 23 we know are your autosomes, or first 22 rather, are your autosomes. And in the case of autosomal monosomy, it means that, let's say for chromosome 1, you only have 1. That is an example of autosomal monosomy. 1 of one of the chromosomes, specifically in this case, the autosomes. So anywhere from chromosome 1 to chromosome 22. Autosomal trisomy is different. So what we have here, and again, this is the one that's often lethal, having less is worse than having more. Autosomal trisomy, tri means 3. So we're talking in this case about having 3 of one of the autosomes. So one of the autosomes between 1 and 22 has 3. These are more common examples. Trisomy of the 13th chromosome, trisomy of the 18th chromosome, or trisomy of the 21st chromosome. This being one most people have heard of. Trisomy 21, so this would be trisomy 21. The 21st chromosome, which is an autosome, has 3, whereas the rest are fine. This is Down syndrome. So while people can survive with autosomal trisomy, it's usually not without some degree of issue. Okay, next up are sex chromosomal aneuploidies. So we are just talking now about the sex chromosome. We talked about the autosomes, the other 22. Now we're talking about the sex chromosome. Sex chromosomal aneuploidy is an abnormal number of sex chromosomes. It can either be more or less. So we have some examples here. Trisomy of the X chromosome. What that would mean is that this would normally be female. If a female is born with a third X, it's usually inactive. So it's usually not a major concern. However, if this would be normal, but instead somebody only has one, this is called Turner's syndrome, and it affects females. So a female that had two and now has one has Turner's syndrome. That's monosomy, one of the sex chromosomes, in this case, X. And this has various clinical issues. Another example would be, let's say you have a male. This is what should be normal, but instead they're missing the X and all they have is a Y. Remember, the Y has very little genetic information. And so because of this would be considered lethal. The underlying cause of most numerical chromosomal aberrations, such as the ones that were described, is non-disjunction, which will be talked about next, and this occurs during meiosis. So let's talk now about non-disjunction. Disjunction would have been a concept you would have learned about a little bit in normal anatomy. And now we're going to talk about this not occurring, so non-disjunction, which is the main cause of aneuploidy in particular. Non-disjunction is an error where chromosomes fail to separate properly during meiosis. So in this normal situation, you can see that, and this is just for simplicity showing you two chromosomes, and what should happen is you should have one going to each. And what this does is it allows for, if this is an egg, for it to fertilize with a sperm, and then you have, let's say this is chromosome 4, you have a chromosome 4 from mom and a chromosome 4 from dad. But when disjunction does not occur, what ends up happening is, so this is an example at meiosis 1, so an early phase of producing eggs, where, let's say this is chromosome 4, instead of one going into each egg of chromosome 4, 2 go in one and 0 go in another. And what this means is that particular chromosome, when it continues through to meiosis 2, will have no chromosome 4. And so when it is fertilized by a sperm, the sperm will contribute its chromosome 4, but there won't be anything from the egg. Another example would be that non-disjunction can occur as well in meiosis 2. So we can see everything's split fine for chromosome 4 here. We have one in each egg. But then at the second division, non-disjunction occurs. And what we end up with is two chromosome 4s that end up in this egg and none in this. Now, I gave you the example already of there being no chromosome 4 and this leading during fertilization or after fertilization to monosomy, where there is only one chromosome 4 and that is from the sperm. Then there's the case of trisomy, where there's 3, and that's where the egg that ended up with 2 continues through, fertilized by a sperm. The sperm gives its one chromosome 4, and now we end up with 3. You can see that occurring here. So the summary here, non-disjunction during meiosis, whether it's at meiosis 1 or meiosis 2, leads to abnormal gametes, and so abnormal eggs in this example, and once it's fertilized by a sperm, these errors become fixed in every cell of that developing organism. And so I gave you the example of eggs versus sperm, but we could flip that around as well. Now let's apply what we've learned about aneuploidy and non-disjunction to the most common clinically recognized example, which is Down syndrome, also known as trisomy 21. So this is where we're looking at an example like this, except in this example of Down syndrome, this trisomy is of the 21st chromosome. So that's the 21st chromosome, or would be if we apply it to this image. Down syndrome occurs when a person has 3 copies then of the 21st chromosomes instead of the usual 2. And you can see that that's represented in this karyotype image here on the right. If we find chromosome 21, you can see there are 3 copies of it compared to 2 copies all around. And just in case you're wondering why chromosomes can sometimes look a bit different in some images compared to others, that is what you might recall the sister chromatids that are connected together before they're pulled apart. But you don't have to worry about that. So how common is Down syndrome and why is it important? Well, as already mentioned, this is a very common example of aneuploidy, but it's one that's compatible with life. It occurs in roughly 1 in 800 to 1 in 1,000 live births, making it extremely important in both medicine and pathology. It's also associated with a significant impact on pregnancy outcomes, with about 75% resulting in miscarriage. And among live births, there is an increased risk of childhood morbidity and mortality. The most common cause of Down syndrome is what we spoke about on the last slide, which is non-disjunction. In fact, about 97% of cases are due to non-disjunction, particularly with the egg. So that would be then maternal. So it's mostly maternal non-disjunction. Okay, so I mentioned that this is most often occurring in the egg, not the sperm. So in other words, this offspring here has 2 chromosome 21 from the mom, which so this would be in this example, this would be the egg. And when fertilized by a normal sperm, it contributes 1, leaving 3 of the 21st chromosome. So when you see or hear about trisomy 21, you should automatically think there was a meiosis error resulting in non-disjunction and that it's usually maternal in origin. Now looking at maternal age. So this brings us to really one of the most tested concepts relating to maternal age and risk of Down syndrome. The risk of Down syndrome increases significantly with maternal age. So here's maternal age and then risk along the side. And you can see, the risk is steady here, but you can see now the sharp incline. And this occurs approximately after the age of 35. So why does maternal age actually matter? Because we're talking maternal age, not paternal age with this risk. If you look at the meiosis timeline here, female eggs actually begin the meiosis before birth. So a female baby in utero has already started the process of meiosis. Okay, so it starts before birth. And then they pause in this suspended state, sort of held in limbo until ovulation later on in life. You can see it if you look close. You don't have to know all the details of this diagram, but just knowing that it starts before birth and then it pauses until the reproductive years. So this means that an oocyte that is ovulated at age 40, so a 40-year-old woman that releases an egg, it had been paused for decades. So this long pause period or arrest period likely allows for errors to accumulate within the sort of cellular machinery that's involved in chromosome separation, and that increases the risk of non-disjunction. So to summarize that, the longer meiosis is paused, as we see occurring with women, with females, the greater the risk of chromosome separation errors. Now let's have a look at some of the clinical characteristics that is associated with Down syndrome. Clinically, Down syndrome is associated with a recognizable pattern of features. And the major ones known include intellectual disability, and this can be of variable severity, hypotonia. So these infants will, say if you have an infant with Down syndrome, they'll appear to have this low muscle tone and shorter stature. That's low muscle tone. There is an increased risk, and this is clinically very important, an increased risk of congenital heart defects. Congenital means born with. So they have a greater chance of being born with a heart defect, especially septal defects. And that's so relating to some of the walls in the heart that keep blood contained within certain chambers. And so this right here is a pretty big factor when it comes to morbidity early in life for these affected individuals. There's also altered immune function, meaning immunosuppression, suppression of the immune system is common, and because of that, there's an increased risk of infection. There's also an increased risk of malignancies, particularly, and that means cancer, particularly leukemia, and more common in childhood. And finally, Down syndrome is associated with an increased risk of Alzheimer's-like changes that occur later in adulthood, often earlier than typical. So sometimes after around the age of 40, there can be Alzheimer-like changes evident in this population. So pertaining to what's in brackets here, whether there's an Alzheimer gene on chromosome 21. So the idea here is that chromosome 21 contains genes involved in amyloid processing, which relates to Alzheimer's, and that will come up in later lectures. So having an extra copy of a chromosome, in this case 21, increases gene dosage and can contribute to early or earlier than normal Alzheimer type pathology. So I've spoken quite a bit about errors in chromosome number, and now we're going to look at structural chromosomal aberrations. So that just brings us back to this. We talked about number, now we're going to look at structural. So of course, it means that the number of chromosomes is normal, but the structure of the chromosome is altered. The two main types shown here are deletions and duplications, but then we'll also talk about inversions and translocations on the next slide. Starting with deletions, just as the name suggests, this is loss or deletion of a segment of DNA due to a chromosome break. A broken chromosome. Now, because genetic material is missing, it should probably make sense to you that deletion of a chromosome, or loss of part of a chromosome, I should say, will often have serious consequences. And a classic example of deletion on a chromosome is deletion on or part of chromosome 5, and this is known as Cri-du-Chat syndrome. And this is associated with low birth weight, intellectual disability, as well as microcephaly, so small brain. And the severity in this comes from the fact that the person has lost or had deleted essential genes within that chromosome. So it'd be like, let's say, here's a chromosome, and this chunk here gets deleted, and then we're just left with this. What about all the important information that was contained here that is now deleted? Okay. So in really contrast, we have duplications as well, and this involves gain, so opposite, gain of a DNA segment, meaning that extra copies of certain genes are present. Now, while this does still disrupt normal development, Duplications are considered to be overall less severe than deletions. So we talked about this before, that it's better to have more than less. I did want to mention that with duplications, while you may see overall less serious effects, it can be problematic then with this person affected by duplications to then have offspring who can end up themselves with deletions or duplications. So a pretty simple explanation on the slide. If this is the original chromosome here for simplicity, and these are all different genes or segments of the DNA. With deletion, you can just see that BCD was removed, and now overall it's shortened. With duplication, you can see that we've duplicated BCD, so we have BCD, BCD, so additional material. The next two structural abnormalities are a little bit different from deletions and duplications because in many cases there's no net loss or gain of genetic material in these examples. And so we can describe these examples as balanced rearrangements, as you'll see when we look at the images here. First of all, inversions. These occur when a chromosome, it breaks in two pieces, and then the segment sort of flips around before reattaching in the opposite orientation. So it's like it's taken apart and then put together back together wrong. So the genes are still there, but their order has changed. So let's look at the example here. BCD it's broken here, and then when it goes back in, it went back in reverse order, DCB. So the genes are still there, but their order has changed. Now, oftentimes individuals with inversions are what we call phenotypically normal. That means physically they look normal, but gene expression can be altered in some cases due to what's called position effect. So we've changed position, right? They're in a new position, which can change how the DNA is read and how products are made from that DNA. And the reason for that, and the idea behind position effect, is now these genes, let's say for simplicity, gene D, is in a different position than it was before. And now it's going to be influenced by A and vice versa. So influence from its neighboring gene that is different than normal. Because sometimes genes that are found beside other genes behave as sort of regulatory agents. And so now if we've moved the position, you could see hopefully how that might affect how overall it's expressed. And because I've used the term gene expression a couple of times already, just to get a definitive explanation of what that is, that's a fancy term to just describe the process by which the information in genes is used to make a functional product that's usually a protein. The bigger concern with inversions is actually during meiosis. So let's say inversions exist and then meiosis is occurring. When chromosomes are trying to pair, the abnormal alignment can occur, increasing the risk of producing gametes that now have duplications or deletions. So a person has inversions and then in the process of producing gametes can produce duplications and deletions. And then if they have children, then the effects can be even worse. Next are translocations. Translocations occur when a segment of one chromosome breaks off and then it decides to attach itself to a different, so we could say non-homologous, it's a different chromosome. There are also reciprocal translocations where both chromosomes break. So you have two chromosomes that break and then they exchange material. Now, if no genetic material is lost or gained, then the individual can appear completely normal. Let's look at how that looks to make more sense of it. This is where images are oftentimes helpful. So this is reciprocal translocation. So it's affecting two different chromosomes. So let's say this is chromosome 2 and this is chromosome 7. Chromosome 2 breaks, chromosome 7 breaks, and then they exchange material. This is what they look like normally, but when the material is broken, so let's say the break occurs here and here, you can see that if the break was here, ABC breaks off, KLM breaks off, so it breaks off right here, and then they go back together, but in the wrong place. So the ABC that was here joins in on the part that broke off here, and the KLM that broke off here instead joins in with the other chromosome. So here's one chromosome, here's another. Completely different chromosomes, so they're non-homologous, so they're not paired. They end up breaking, and then the piece that's broken goes back to what it thinks is the right place, but it's on a different chromosome. Now, similar to inversions, problems often do arise during gamete formation, where this unbalanced chromosome segregation can lead to embryos that are missing or have extra genetic material. So the key idea here is that these balanced rearrangements may be silent to the individual, but they can significantly increase reproductive risk, which is why they're clinically so important. So you can have somebody that has translocations that exist and they don't know it, and then they decide to have a baby, and then that genetic mutation that they had ends up passing on to the baby and being worse. So let's continue on looking at another example of translocations. With this translocation, and this is going to be an example of reciprocal translocation, remembering that if it's just translocation, you have one chromosome that's broken and it becomes part of another chromosome. But with reciprocal, two break and then become part of the wrong chromosomes. They just trade places. So here we have a normal chromosome 9 and normal chromosome 22. So if we were looking at a karyotype, normal chromosome 9, normal chromosome 22, but we're just looking at one of them for simplicity. And what we end up seeing here is that a break occurs here on chromosome 9 and a break occurs here on chromosome 22. And then in the new example, you can see based on the color differences that they've traded places. So this ends up joining in here, and this break ends up joining in here. Now, when we look specifically at chromosomes 9 and 22, where they swap places, the result is a chromosomal defect called Philadelphia translocation. So I'd like you to make note of that. Philadelphia translocation, or sometimes written as Philadelphia chromosome. And what's important clinically here is that this can be seen in those that have leukemia, particularly a leukemia called CML, or chronic myelocytic leukemia. So it's really cancer of the white blood cells. Now, just to be more specific on that for you to make note of, what really is the Philadelphia chromosome? It's the new chromosome 22. So this is the new chromosome 22 that you can see is now called Philadelphia chromosome, which is seen in people with chronic myelocytic leukemia. All right, that's the end of this lecture. Thanks a lot for your time.